Danny is a UT Austin PhD graduate. He got his PhD in chemistry. He worked in Andy Ellington's lab and also worked with Eric Anslin. And we discovered him at the machine learning lab as someone who was trying to do machine learning but didn't really know what he was doing. No, just kidding. I'm just kidding. Actually, a very talented, a very rare, very rare person who's both, very talented at machine learning and also very talented in his, has all the domain expertise. One way to get Danny going is to ask him like a high school chemistry question that you never understood, and then several whiteboards later, you'll still be confused. But I think it's fun, interacting and working with Danny. He's now an IFML postdoc. We've hired him to sort of lead our AI for protein engineering group. And, it's endlessly fascinating talking with Danny because you realize, how cool the natural world is from both a chemical and biological perspective, and how even the smallest reactions can be really, really surprising. So he's going to talk about some of that work today. And then, thank you for being here.
Well, thank you guys for having me here today and coming to hear me talk a little bit about the work we do here at Deep Proteins. I'm really excited actually about this talk because it's the first time we disclose this project. So some of you guys have seen me talk before, and it's always kind of been like similar work, and then we just kind of incrementally add or do new things in it or make a new protein. But this is a brand new project.
You never want to refer to your own work as incremental.
I have self-awareness. But I think this is one project I'm really excited about because I would say it's far from being incremental. So I'm happy to share it with you guys. So today I'm going to talk to you guys of how we've been learning how evolution engineers proteins and trying to leverage how evolution engineers proteins so we can engineer proteins. I know not all you guys are machine learning protein engineers, so I'm kind of going to start at the very beginning. And that is, what are proteins? So in simplest terms, proteins are the workhorse of biology. They essentially do everything for biology. And thinking about it from like a machine learning LLM perspective, we just have a vocabulary of 20 characters or 20 words. And these are these 20 amino acids. And the 20 amino acids are, some of them are similar to each other, most of them are different, and you have these different groups. And the grouping is kind of showed by how they're binned in these colors. And then whenever you polymerize these amino acids or put them into a string together, that unique string or the order of these amino acids will fold into a unique 3D structure. So you can kind of see it over here. We have the primary sequence, which is just a string. This primary sequence will lead to what is called secondary structures, which are usually shown as these helices or these sheets, these arrows that are kind of next to each other. And then these helices and sheets and then unstructured loops come together to form what is called the tertiary structure, which is just one folded protein string. And then multiple protein strings can come together to form what is called the quaternary structure. So you can see that here where you have multiple strings shown as different colors and they come together. And the terminology here can get a little confusing because like the tertiary structure could be called the protein, and the quaternary structure can also be called a protein. And some proteins only have tertiary structures. Some proteins only work as quaternary structures. But in general, some folded up amino acid strings, that's what a protein is. And the way it kind of works is these amino acid sequences will fold into 3D shapes, and then it's the specific 3D shape it adapts that endows a protein with its function. So as you change the 3D shape, which means you change the sequence, the protein's functions change. And proteins, like I said, are the workhorse of biology. So they do all the functions or everything we see in biology. So to give you guys some examples, one would be this protein here, which acts like a pore, similar to whenever you're grating cheese. And my favorite protein here is this ATP synthase, and it works kind of like a turbine. So kind of like how a dam, water flows through, causes the turbine to spin to generate electricity. We have a very similar protein where there's a gradient forms of protons, and it flows through, causing this to spin. And this is how we generate energy in our body. So this protein right here spinning is the primary reason why we all have to breathe. So if you want this protein to quit spinning, it's really easy, just stop breathing. So now that we kind of know what proteins are, well, why do we do protein engineering? So natural proteins, which are everywhere, they often need to be engineered if we want to commercialize them or if we want to do something with them in the market. And the main reason why is because nature optimizes proteins for whatever nature wants to do. Proteins aren't necessarily optimized for what humans want to do. So therefore, that usually manifests itself as they being like not sufficiently stable or their function just isn't good enough to use it in whatever applications we're interested in. So protein engineering is the field of actually taking these strings of amino acids and identifying positions in the strings where we can mutate the amino acid to another amino acid. So just to describe a little bit of jargon here, a residue is essentially adding what amino acid is located at a particular index of the string. So it's location and identity. While wild type is the identity that nature put there, and a mutation is whenever we change that identity to something else that's not found in nature. So you hear me use these words, and that's essentially what they mean. And so here's some examples of what an engineered protein might look like, where you start off with some natural protein, and then you can see all these positions in the string, which are all the colored blobs that you see everywhere, like the pink and the purple, are where we actually identify positions in the string and we want to change them to something else to improve some function, whether it's shelf stability, temperature stability, pH, activity. So now that we can understand what protein engineering is, why is this kind of important? Well, the big area where we engineer proteins is to develop biologics to treat diseases. So currently, biologics account for a $400 billion market. Over 50% of current cancer drugs are protein-based drugs. And the reason why is because proteins tend to be highly specific, which means you reduce the off-target side effects that you get from these drugs. And they actually can target a lot of other things in biology that small molecules can't. However, developing biologics is very hard because they're aggregation prone. You can't usually make enough of the protein for it to be commercially viable. You can also be allergic to these biologics. And they're just difficult to engineer. Another cool application of, one cool biologic that actually came out of UT was the COVID-19 vaccine by Jason McClellan here in NBS. He identified a few mutations on the protein that enabled us to stabilize it so you can actually mass produce a vaccine from it. And here's one of my favorite quotes on protein engineering. This was from the head of computational protein engineering at Roche. He says, we get the starting point from nature, which provides an optimized framework, and then we endow them with superpowers. So it's really cool to think of yourself as a protein engineer as someone who gives protein superpowers. So this is kind of like traditional protein engineering. They've been doing this in pharma for, I would say, 20 to 30 years now. But there's actually new emerging areas where protein engineering is actually starting to make a big impact. And that is in creating a sustainable future. So I would say that one of the first big success in protein engineering was in pharmaceutical manufacturing of insulin. In 2021, they used it to biomanufacture molnupiravir, which is one of the first antivirals to treating COVID-19. So if you didn't get the vaccine and you actually got sick, this was one of the first drugs approved to treat you. And it's purely made where they just ferment sugar and make this drug because it's all done with proteins. There are currently a lot of cool reports coming out on developing enzymes for making cancer drugs, also for actually making new types of antibiotics. And on the non-pharma side, recently there's this big company coming out. They came up with a protein to biomanufacture hydrogen peroxide. LanzaTech was able to do carbon capture to produce ethylene, and ethylene is used to make lots of things like plastic bags. LanzaTech's also worked on generating jet fuel from just waste gases. So they take you can think of gas that comes out of like a car exhaust, and then they can return that into fuel. Even Lululemon's getting in the game, where they started using these plastic-eating enzymes to recycle textile. And actually, a few weeks ago, they just launched jackets where they used an enzyme called PETase to take water bottles and then produce jackets from water bottles. And actually, one of my favorite applications of proteins in the food industry is they have figured out how to take the same PETase enzyme and produce vanilla flavor. So we're turning water bottles into vanilla ice cream. So protein engineering is actually a big growing field. So now that I kind of showed you guys why we care about protein engineering and its potential, I'm kind of going to show you why it's hard. So I kind of alluded to this earlier, but just identifying how to mutate a protein is very difficult. If you have a protein of length n, there's 20 to the n possible sequences you can make. And most of those sequences are actually dead proteins. They have no function. So it's a very sparse space. The average length of a protein is about 300 amino acid. So that would mean you can make 5,000 to 700 single point mutations on that 300 character string. And if you wanted to make double mutants, so change 2 of them, there's over 16 million combinations. So, and most of them actually don't benefit the protein. Most of them actually kill it. So how do you identify this small subset that actually improve the protein? Because identifying them is very expensive. It costs a lot of time and money. And if you only get so many shots on goal before you run out of money. So it's the whole field of protein engineering has been trying to figure out how do we efficiently search this space. And I would say over the last 20 to 30 years, computer-aided rational design, these are primarily like physics-based methods, and directed evolution have kind of dominated the space. Directed evolution is where you pick a subset of the protein, you randomize it, so you make a library, you screen the library, hopefully something is better, and then you start from that as your starting point and then kind of repeat the cycle. While rational design is very similar, but instead of making a library, you use some intuition, some background, or some computer program to help narrow the space down for you. So this is the Center of Generative AI Symposium. So this is a great point, great spot to transition to machine learning and how machine learning can actually guide protein engineering. So a little background on machine learning guided protein engineering. Whenever we need to engineer a protein, we often need to optimize multiple functions. It's usually not just one thing. So here's just a handful of the functions, but definitely not all of them. But we often need to stabilize the protein, improve binding affinity, or improve its activity, or improve its expression, how much we can make. And unlike in natural language processing or like in computer vision, there's just a lack of experimental functional data sets to actually guide and do supervised learning for these functions. There is no ImageNet like there is in computer vision or glue in natural language processing. I would say the one exception here would be AlphaFold because they did supervised learning on protein structures or the PDB. But this, looking at kind of like how proteins work, right? We have sequence, structure, and function. We have very little data in function. AlphaFold was going from sequence to structure. And here's kind of the amount of unlabeled data that we have. So on sequence space, we have about 250 million sequences, but only about 0.3 of this space has been experimentally annotated. So very little labeled data. And for structure space, we have about 125,000 of unique protein sequences where we've actually solved the structure. So in protein engineering, most deep learning frameworks utilize a BERT style or masking type of pre-training. So in sequence models, this is just traditional BERT. We pass in a sequence, we mask an amino acid, and then we try to predict what was masked. The work that we've been working on is primarily on structure-based models, where we use these microenvironments, but it's the same thing as BERT-style training, where we select a residue to mask, shown here in pink, but we just extract the local chemistry surrounding it, and then we delete what we centered on. And then there's a second modality in structure-based machine learning called inverse folding, where you take the whole structure, you keep just the backbone, and then you try to predict from the surrounding backbone nodes what was masked in the center. So the key distinction between these three data modalities is that sequence models like ESM2, they use the global context or the full protein sequence to predict what was masked, while computer, the microenvironment modality, is at the atom level. You get local context, and protein NPNN, or this inverse folding framework, they look at amino acid level, but it's still a local context. So we've demonstrated that these self-supervised pre-trained models can actually, just by zero-shot training on unlabeled data, can guide protein engineering projects in the lab. So the first protein we made was a fluorescent protein. We made a protein for COVID-19 diagnostics during the pandemic. We made plastic-eating proteins. This is actually the protein that they use to eat the plastic to then turn it into vanilla. This one was one that we recently published, where we were able to combine AlphaFold and these self-supervised pre-trained structure models to do active site engineering of an enzyme for a pharmaceutical manufacturing of galantamine. We've also been using them to stabilize and improve developability properties of this cancer therapeutics. But when we first started working here at IFML on this problem, we wanted to figure out, how do we improve training these models, right? We had this traditional masking self-supervised learning, and we were then evaluating them on downstream data sets of experiment of stability. And one thing we realized is that the whole community uses wild type accuracy, or how well you predict what you masked, similar to NLP, as a measure of improving the model. So for example, here in blue is this LLM out of Facebook, ESM. It can recover what was masked 95% of the time. But its ability to actually predict gain of function or stabilizing mutations is really low. And while our model was only 84, it wasn't much better. But we realized that if we actually used a model that was much worse at predicting wild type, so the wild type accuracy goes down, we actually get better performance on engineering proteins. So you can see our F1 scores goes up and our recall goes up. So we realized that optimizing the wild type accuracy actually hinders your ability to engineer proteins. So there was this disconnect between the metric used to improve the models, and then actually the model's ability to actually guide protein engineering downstream. So we did some further experiments to see this relationship, and we see that as we train the model in these self-supervised fashion longer, we see that the wild type accuracy continues to increase, right? We can nearly match a large language model's performance, which was 95. We're hitting 92% accuracy. But as we keep improving the wild type accuracy, we see its correlation with experimental data actually starts going down. So this is actually not known really in the field, and most of the machine learning guided protein engineering community is always trying to improve the wild type accuracy. And they'll actually report where they were unable to make it better. So their model actually did not beat baselines, which is the wrong metric to evaluate if you made any meaningful improvements on your model. So we started thinking about, okay, well, how do we generate a self-supervised learning task that actually, if we do that, where we have a metric where we can actually improve the model, it actually leads to improved protein engineering. So that's when we started looking to nature for inspiration. And we started looking how evolution engineers proteins. And we came up with using evolution-based soft labels. So before, we would just mask an amino acid, like what's shown here in orange. And because we know what we deleted, we knew what the answer was. So that would be like a hard label classification problem. Instead, what we did is we just took this protein and its entire family. So you can think of like its cousins, its brothers, grandma, and we align their sequences and then look at the diversity at that position. And then we just turn that diversity into a distribution. And then now, rather than predicting what we deleted, we actually try to predict the distribution that's observed here in nature. So we essentially changed the loss from going from a hard label to an evolution-based, sorry, my mouse isn't working, evolution-based soft label. So when we compare these two models, we can see that while our wild type accuracy no longer continues increasing up to like 92% and it starts plateauing, we actually also plateau and don't actually lose performance as we keep improving the model. And we actually get a slight bump in improvement, where before our best correlation was 34%, a Pearson correlation of 34. And now we're getting Pearson correlations of up to 0.37 on this exact data set. And then whenever we compare it against baseline models, so against a sequence-based self-supervised model, an inverse folding-based self-supervised model, our microenvironment-based self-supervised model, and then now our soft label self-supervised model, across different phenotypes or protein functions, we have 3 for binding affinity, where these two are how well do 2 proteins come together, while this platinum DB one is how well does a small molecule or a drug come together with a protein. Yeah. Yeah. Yeah, so there's 2 ways of doing that. The most popular way would be using sequence similarity, because the protein's a sequence. So you can measure how different 2 sequences are, and then use like a sequence threshold cutoff. And also the other way is you can just take these sequences if they have structures, or if there's an alpha fold structures, you can align their structures and then measure like a distance between the atoms. Any other questions?
When you say align the structures, you mean like test rotation one versus the other?
Yeah, essentially how do you, what's the best way to superimpose the backbones of these proteins where you minimize the distance between the atoms? Yes and no. So the structure-based alignment, those are a lot of brand new tools have recently been coming out. Sequence-based alignment tools, at least tools that can scale to massive data sets have been recently coming out. Tools for sequence-based ones are much more mature and well-established. So people primarily use sequence-based, but we also kind of understand the relationship between protein sequences and protein structure. So there's this phenomenon called the twilight zone. So once you kind of go below like a 30% sequence similarity, so like between 30 and 15, 30 and 20, the structure dramatically changes based on the sequence. But if you're above this zone, so if you're like at 35, 40, 45 sequence similarity, you're like very confident that they were going to have similar structures. So usually we use like a 30 to 20% sequence similarity as our cutoff, because we know we have a high confidence where all the proteins above that will have the same structure. So therefore we can make meaningful alignments. Any other questions? So where was I? Yeah, so we have a few binding affinity data sets and then a few stability data sets. Here, stability means if you mutate the protein, will the protein no longer fold? Or how stable is this folded structure? How much energy do you have to put into the system before it falls apart and becomes a noodle? So you can see by us moving from hard labels to soft labels, we made some meaningful improvements from the green to the blue across these data sets. But we're not that much better than, I would say, like the best model, right? It was just marginal or incremental improvements whenever you do zero-shot prediction with these models. And this is on regression metrics or a classification metrics. So after seeing these results, we thought, could we do better? There's so much information in the diversity that we observe whenever we align these proteins, where we felt like we weren't squeezing it all out. And so that's kind of what led us to what we call evolutionary ranking, or EVO rank. So EvoRank is, I would say, two fundamental changes in how we view the problem. And it stems from the fact that we know that protein engineering is about identifying amino acid substitutions, where a substitution means mutating from an amino acid to an amino acid. So instead of actually trying to predict the distribution that we observe, can we reformulate the problem as a ranking problem, where if we give you two amino acids, can you rank which one is better than the other? So essentially, we can take this MSA distribution that we observe at a specific column and then turn this into a continuous value where we take the log odd between the to amino acid and the from amino acid, giving us what we call an EVO score. And then instead of having the model take in the masked microenvironment and then just give us the likelihood distribution of all 20 amino acids, we can actually take the masked microenvironment and explicitly tell it what amino acid we want to mutate from and what amino acid we want to mutate to. And then given the local structural context and what I'm mutating from and to, give me a rank score, or like give me how much better is one over the other. And then now we can train the model as a regression model using a mean squared error as our loss between the evolutionary score and our model's predicted rank score. And this architectural design was actually inspired by one of our previous projects, Stability Oracle, where we first came up with this graph transformer architecture. And you can see it here in the regression head, where we actually take a from and a to, contextualize them, and then turn it into a phenotype. So here, instead of predicting something from in a supervised fashion from a data set, we're actually doing self-supervised learning where we're predicting what evolution would do here. So again, comparing the hard label classifier to the EVO score regression model, you can see that our wild type accuracy plateaus around 62, but we get significant improvement in our correlation with the stability data set, and we don't see this drop in performance anymore. And then when compared to all our baselines, we see that we get massive improvements compared to our previous self-supervised models, where I think for regression task, it's on an average like a 40 to 50% improvement. And it actually bridges the gap whenever we take, so you can take all these self-supervised models and then fine tune them on a downstream task. So that would be their supervised analog, I guess you would call it. And you can see our self-supervised method, EvoRank, actually bridges the gap between the self-supervised models shown as circles and their supervised versions shown as stars, where you can see our self-supervised model, MuteRank, is more similar to the supervised models than its predecessor, wild-type masking self-supervised models. And you can see there's a clear gap between the two. So because we can take these self-supervised frameworks and fine-tune them on smaller experimental data sets, we then evaluate how an EvoRank pre-trained model can be fine-tuned in this setting. So you can see we have this cDNA data set that was recently published in Nature. It's the biggest protein experimental data set to date. We have a subset of it that has about 117,000 single point mutations spread out across about 116 proteins. So what we did was we partitioned this data in two different ways. One is, what if we just grab a subset of the mutations at random? So 11,000 randomly, 54,000 randomly, or take them all randomly. Or what if we partition the data based on the protein that they're located on? So if we grab all the experimental data for 10 proteins, or all from 50 proteins, or all from 116 proteins. You can see here, when we do the fine-tuning, that if we fine-tune with about 11,000 mutations, the previous fine-tuned model still can't outperform our zero-shot model. And if we are the EvoRank fine-tuned model with only 11,000 mutations is nearly already matching the performance of fine-tuning on all the data. So we're getting essentially this similar performance, but using 10x less experimental data. And then if we partition it based on the number of proteins we have available for training, it's even more dramatic, where the previous fine-tuned model is significantly behind our zero-shot model. And we're still nearly matching the performance of if we would have fine-tuned on all the data, so using 10x less of the data. So hopefully I've convinced you that our EvoRank-based representations are more expressive for protein engineering applications. And we're actually currently working on generalizing our EvoRank loss to other data modalities, such as sequence models to make ESM rank and inverse folding models. That's it.
Thanks, Danny. We have time for a couple questions. You, sir, you seem to know something about biology.
Yeah, so the mutations that you see at a site, we know often depend on mutations at other sites. And I'm wondering whether that is in any way possible to feed into your training. Like the idea with ESM is that somehow the model just figures it out by itself because you always give it one sequence.
Mhm.
And so sometimes you have a mutation in this site and a mutation on that side over there. And so the model sees that and figures it out. Is there any way to put that into your model?
You're saying like an epistatic version of EvoRank.
Yeah, essentially.
Yeah, so we're currently developing frameworks where we're generating an EvoRank score, not just for single point positions, but actually for doubles and triples. We're setting up, that's like active things that we're doing to generalize the method.
The score should depend on like
Yeah.
what exact sequence.
Yeah, exactly. Yeah. So the score should depend on like if it's a double mutation, what actually condition on what's surrounding them and what you're actually trying to mutate to, right? Because if you pick column 1 and column 2 and column 1 and column 4, that should be a very different score. Yeah, so it's not trivial, and there's different ways of formulating that. So we're kind of just setting up several methods of formulating that and just seeing which one works best.
Or I think maybe an additional, maybe I didn't understand your question, but you can, like one thing you could ask is, well, why don't you retrain ESM using like an EvoRank loss, to get the full global scale on this? And that is something that we're working on.
Yeah, that's also something we're actively doing. Mm-hmm.
Let me just get to someone else. Yeah, go ahead.
So I was curious, I'm not super familiar with this problem space, but I know that like certain types of Duchenne's muscular dystrophy can be alleviated with the protein, which has an error in it, being substituted with a much smaller version of the protein that has similar structure and function. And so my question is there any work in this space which is focused more on not just single amino acid mutation, but instead actual cutting down the length of the sequence itself?
Yeah, so that would fall a lot more under like machine learning at like protein design, where like you kind of like diffuse a scaffold, or you can like modify the scaffold or truncate it and then try to generate sequences that will fold into that. That's a whole other area of active research. So yes, but we usually focus on taking things that already kind of work and making them better, because that's easier to kind of get things over the.
Yeah, there's some distinction between sort of protein design and protein engineering,
Mhm.
And I'd say, protein design is more of a de novo design,
Mhm.
Something that's radically different than what we've seen before, whereas
Mhm.
protein engineering is making, a smaller number of changes. Yeah.
Mhm.
I see. Thank you.
Mhm.
Yes.
About with Evo, really nice. With Evo rank, you'll have this system, but then you mentioned these parameters of protein stability,
Mhm.
binding affinity, all these things you maybe desire in your protein therapeutic. Would it be as simple as like, and you mentioned how do you find which proteins you want to test? So down the line for like a company, a biotech company, would they just take, this is the top ranked ones, and we're just going to take the top 2, 3, and then make those proteins and then test it out? Or is there, I guess you're also taking things like, these other desired properties like stability, lack of aggregation. How would you sort of envision that?
Yeah, so essentially what you said is what we have done in the past a lot. So like in this slide, a lot of this came from looking at the top, where we would look at all the predictions that the model made, compute the log odds between the to and the from, where from was wild type. And then once we have that sorted, we would then manually kind of go through the top 10, 20, 30, 40, and then filter out them based on how much chemical plausible sense they make, but also where they located. Is there some information the model does not know that the human does know? So for example, if it's like a protein therapeutic, we probably don't want it to be on the surface because then it could become immunogenic. So we would kind of try like excluding them based on their location. So there's a lot of post-processing to filtering down what the AI thinks into what people actually go and make in the lab.
Yeah, okay. So just a quick question, like, why do you care about zero-shot accuracy after pre-training? Can you not fine-tune for your actual downstream task?
Most of the time we cannot. Yeah, so
I mean, to fine tune, you need experimental data. And to get the experimental data that we need, like a deep mutational scan, is very expensive and very time consuming. And so it's just very difficult.
yeah, like in my experience, when we give somebody like, let's say 20 mutations to go make, we'll get data back anywhere from 3 to 6 months. So we don't have the 20 or 100 to start with, even to begin fine tuning most of the time.
Thanks, Danny. Great talk. You listed several problems with biologics manufacturing right now, and I'm curious what you think the research community can do to have the biggest impact on actually accelerating biologics manufacturing. Maybe it's protein engineering or protein design or something else.
Yeah, I would say pretty much all biologics that end up getting into the clinic, and then if hopefully they get FDA approval, those are all engineered biologics. Like they didn't just come naturally out like that. Most of the time they'll come from like mouse or rats or like animals, and then they have to humanize them. Or there's just lots of different things that they have to kind of do to get them to work and play well as a therapeutic. So what I think would be most helpful would be, wow, if companies, whenever they go through this whole process, if they actually provided all that experimental data, all those trial, and because it wasn't like a one step, like here's natural, here's our variant that we're putting in the clinic. They like had to iterate, make mutations, try different things, see what worked, see what didn't work, and then that data just never sees the light of day. I'm just saying, like, if that data was available, then we would be able to do more machine learning. Oh no, I completely understand why they don't. I'm just saying, like, if you would have asked me what is needed, it's data. I'm not telling them to do it. I'm just saying, like, that's what we need,
Okay, let's thank Danny again.